Okay, thank you very much, Gary. I'll just share my screen now. Can everybody see that okay? I guess I won't get a response, but okay.
Yes. Good.
Okay, great. So as Gary mentioned, today I'll be talking about how we found that there's regulators of chylomicron secretion in both humans and animal models. So this is important because we know that patients with type 2 diabetes and pre-diabetes have elevated secretion of lipoprotein particles from both the liver and intestine. So you've probably heard of LDL cholesterol, which is considered the bad cholesterol, and this is a remnant particle from the liver lipoprotein particle, which is VLDL, or very low density lipoprotein. So as TG gets removed from this peripheral tissues, you end up with this remnant particle. And the same thing happens with chylomicrons that are released from the intestine. So chylomicrons from the intestine deliver dietary fat to peripheral tissues. And as those tissues pull out TG, you're left with this chylomicron remnant. And these remnants and LDL particles can cause problems because they can penetrate the endothelium of blood vessels and form atherosclerotic plaque, and that can lead to heart disease. And we know that this is particularly an issue in people with type 2 diabetes or pre-diabetes because they have an increased burden of these remnant particles. So we know that chylomicron secretion from the intestine is primarily determined by how much fat you eat, but we've also seen over the last few years that this is fine-tuned by nutrients, hormones, and neural signals. So what I'll talk about today is how nutrients and hormones fine-tune chylomicron secretion. So there's chylomicrons and VLDL coming from the intestine and the liver. The lipid within chylomicrons is coming mostly from dietary fats, but also from free fatty acids that are released by adipose tissue. And those free fatty acids can also go to the liver and become incorporated in VLDL. What we found is that there are regulators of this. So acutely, insulin coming from the pancreas inhibits chylomicron and VLDL secretion. But when there's insulin resistance, you have less of this inhibition. And so that's contributing to increased chylomicron and VLDL secretion in that state. Glucagon also inhibits VLDL production, which is something we've studied in humans. The intestine itself also secretes hormones, so it secretes this glucagon-like peptide 1, GLP-1, and glucagon-like peptide 2, or GLP-2. And these then act on the intestine to regulate chylomicron secretion. So we've shown that GLP-1 inhibits chylomicron secretion, whereas GLP-2 stimulates chylomicron secretion. We've also looked at nutritional factors like glucose and fructose and found that both of those stimulate chylomicron secretion. So what I'll focus on today is what we've seen with glucose and GLP-2 and how we've figured out how these actually stimulate chylomicron secretion. So they both do stimulate chylomicron secretion, but they're doing this by different mechanisms. So I'll talk about how we've evaluated that in humans and in animal models. So just briefly, I want to go over how chylomicron secretion occurs. So we have these enterocytes in the intestine that have these microvilli that face the intestinal lumen. So dietary triglycerides are hydrolyzed to monoacylglycerol and fatty acids, and these are then reesterified into triglycerides at the ER. The majority of these triglycerides end up being packaged into these pre-chylomicrons and transported to the Golgi and pre-chylomicron transport vesicles, and then are secreted out the basolateral side as mature chylomicrons. When they're secreted there, they pass through this lamina propria area to get into the lacteal, which is this blunt-ended protrusion off the mesenteric lymph duct. So the mesenteric lymph duct then pumps these chylomicrons into the circulation. Alternatively, some of these lipids end up being stored within the enterocyte as cytoplasmic lipid droplets. So we can see in humans, when we've done biopsies, we can see these lipid droplets in enterocytes up to 10 hours after a high-fat meal. And we can also see that there's some chylomicrons that stay sitting in the lamina propria or in this intracellular space. So they stay there kind of stored within the intestine, and these can be triggered to be secreted as chylomicrons many hours after a meal. And what I'll show you today is that we think that glucose primarily acts on mobilizing these cytoplasmic lipid droplets, whereas GLP-2 seems to be acting extracellularly, so beyond the cell, and mobilizing chylomicrons that are sitting either in the lamina propria or in this intracellular space. So for the first part, talking about how oral glucose mobilizes intestinal lipid stores, we studied this in humans with two different protocols. So in the first case, we did a plasma sampling study, which was a crossover design. So we had healthy individuals. They came in the morning and had a high-fat liquid meal of 50 grams of fat. And then 5 hours later, they drank either glucose or water. So they came in for 2 visits and had either glucose or water on each of those visits. And we followed them for an additional 3 hours, taking blood samples. So the idea here is that when they drink that high-fat drink, the TGs will go up and they'll start to stabilize by 5 hours, but we'll still have some of those intestinal lipid stores, and we want to see if glucose or water glucose will mobilize that compared to water. In a separate study, we did similar timelines, so high-fat drink, then glucose or water at 5 hours, but this time an hour later we're taking a duodenal biopsy. So these are subjects that are already going to undergo gastroscopy for clinical reasons, so we recruit them to the study so that we can also take duodenal biopsies while they're undergoing that procedure. So for the first part, we see that after you drink a high-fat drink at time 0, your plasma TG increases up to 3 hours and then starts to decline. But if you drink glucose at 5 hours, you see an attenuation in that decline. So there's actually a stabilization of plasma TG for a few hours after drinking glucose. And we think that's predominantly due to this increase in chylomicron TG when you drink glucose at 5 hours. Afterwards, chylomicron TG increases. But there's no increase in VLDL TG. So this suggests to us that 5 hours after fat's ingested, glucose can mobilize that fat. In the duodenal biopsy part of the study, we could take duodenal biopsies 1 hour after drinking glucose and 6 hours after drinking fat, and look at the enterocytes using electron microscopy and actually visualize these cytosolic lipid droplets and secreted chylomicrons. And we can quantify how big these CLDs are and how many of them are, and try to understand how glucose is affecting these. So this is a similar image. This is actually from mice, but you see kind of the same thing in humans, where you can actually see these cytosolic lipid droplets of various size, and you can see TG within the ER lumen, TG within the Golgi, and within these secretory vesicles. So you can quantify where lipid is and how many of these secretory vesicles there are to try to figure out how glucose is affecting these lipid pools. So this is published work. I'm just going to summarize what we found from microscopy and the plasma analyses. So when subjects drink water and we look at the duodenal biopsies, we see a lot of these CLDs within the cytoplasm of various sizes, and we also see lipid within the ER and the Golgi and secretory vesicles, and then some chylomicron TG still in the blood. But if you compare that to glucose, when they drink glucose, you see less CLDs in the cytoplasm, and the ones that are there are smaller. And there's also less in the secretory pathway. There's more chylomicrons within the blood. So this really suggests to us that in humans, oral glucose is mobilizing those enterocyte CLDs from the intestine. In addition to looking at microscopy, we also did proteomics on those duodenal tissues. And one of the things that stood out to us is this syntaxin binding protein 5. So it had a high p-value and a quite significant fold change from water to glucose. So we see a downregulation in syntaxin binding protein 5 with glucose. And what this protein is a negative regulation of snare protein assembly. So when the pre-chylomicron transport vesicle coming from the ER goes to the Golgi, it forms this snare complex to facilitate maturation of the chylomicron. But this syntaxin binding protein 5 can bind the syntaxin 5 and inhibit this snare formation. And what's been shown in pancreatic beta cells is that glucose induces degradation of syntaxin binding protein 5, and that's thought to be part of the way that glucose promotes insulin secretion. So we think that maybe a similar thing is happening in enterocytes, that glucose is downregulating this syntaxin binding protein 5, and that helps facilitate chylomicron maturation and eventual secretion. So this is really as far as we can go with humans. And so to kind of figure out, is this really happening, and what's the role of those intracellular CLDs, we had to move to animal models. So we decided to use rats to elucidate those mechanisms. We do this through a cannulation surgery model. So on the first day, we're doing cannulation of the mesenteric lymph duct and putting a catheter into the duodenum. So this allows us to continuously collect lymph flow or lymph fluid and calculate lymph flow, look at the TG concentration, and see how those are changing over time. So we do the surgery on the first day, and the second day we try to mimic what we did in humans. So we start at time 0, giving an intraduodenal lipid bolus, and 5 hours later we give either intraduodenal glucose or saline. And throughout this time we're continuously collecting lymph fluid. In a separate study, we did no surgery, just oral gavage. So again, oral gavage of lipid at time 0, oral glucose or water at time 5 hours, and then 1 hour later, taking duodenal tissue. So this is to mimic the biopsy study in humans. And this is really to look at the role of syntaxin binding protein 5. Unfortunately, this part is delayed due to COVID, so I can't give preliminary data, but I just wanted to show that that's how we are approaching that question. In terms of the surgery model, so again, this is published work, so I'm just going to summarize with this cartoon. But what we see is that when you give glucose rather than water, we see an increase in lymph flow rate. So as we're collecting lymph fluid here from the mesenteric lymph duct, we see that glucose increases the flow of lymph fluid with an initial delay that we think is due to the osmotic effect of glucose. And we also see an increase in lymph TG output, and that's the calculation that is lymph flow times TG concentration. So it takes into account those two major factors that are affecting TG output, and we see that there's an increase in TG output. We also see an increase in the ratio of TG to ApoB48. So each one of these chylomicron particles has just one ApoB48 protein on it. And so if there's an increase in this ratio, it's suggesting to us that the chylomicrons are larger, and that's likely due to the mobilization of intracellular lipid stores to allow for further lipidation of chylomicrons before they are secreted. So we think here that glucose is mobilizing intracellular lipids. To ask, is this really the case? We decided to inhibit the Golgi apparatus. So we thought that if we can inhibit Golgi, which is very distal in this secretory pathway, then maybe that will inhibit glucose-mediated TG mobilization if it really is the case that glucose is acting primarily on these cytoplasmic lipid droplets. So we did this by infusing intraduodenal Burfeldin A, starting 1 hour before we give the intraduodenal glucose. So our preliminary data suggests that it is the case that Golgi disruption diminishes glucose-mediated TG mobilization. So we see that when we give Burfeldin A and stain the Golgi here in red, that it causes dispersal of the Golgi, and this has been shown to affect its function, and that it can no longer process these pre-chylomicron transport vesicles. We see that this doesn't affect lymph flow. So lymph flow is basically the same, whether glucose is given in the presence of Golgi disruption or just vehicle. But there is suggestion that it inhibits TG concentration. So we do need to increase the numbers to deal with these error bars, but so far it looks like Berfeldin A mediated inhibition of the Golgi is disrupting this TG mobilization by glucose. So that's all I have to say about glucose. The other stimulant I wanted to talk about was glucagon-like peptide 2. So again, this is a hormone that's secreted by enteroendocrine cells within the intestine, and it can act on neural signals, and we think that that's how it's influencing intestinal lipid stores, and there could be other ways that it's affecting this as well. But we wanted to see in humans first, doing a similar study where we give again, lipid, and then 5 hours later, we're giving either teduglutide, which is the pharmaceutical analog that's used in humans, or placebo. So again, we're doing a plasma study and a duodenal study. So giving teduglutide or placebo 1 hour before to do a duodenal biopsy. In terms of the plasma side of things, we see that GLP-2 rapidly increases plasma TG, so it's given here 5 hours after a lipid drink at time 0. And if you just look at that as a percent of lipid at time 5 hours, we see there is this robust increase in plasma TG with teduglutide. And this is due to a robust increase in chylomicron TG that's reaching a peak by half an hour after GLP-2 is given here at 5 hours. For this study, we did that duodenal biopsy arm as well, and we're waiting on those proteomic and microscopic analysis to look at the enterocyte and see if we can see a difference in those extracellular and intercellular lipid pools. We did the same surgical cannulation model in rats, and in this case, we saw that GLP-2 rapidly mobilizes TG without altering chylomicron size. So we see a robust and very rapid increase in lymph flow that's sustained throughout the 2 hour period after giving GLP-2 or placebo. And we see an increase in lymph TG output. In contrast to glucose, where we saw an increase in the ratio of TG to ApoB48, here we're not seeing any change in the ratio. So this is suggesting that the chylomicrons are not changing in size between GLP-2 and placebo. So this suggests that there's mobilization of these preformed extracellular chylomicrons. And we've done further studies, again, with Berfeldin A to disrupt the Golgi. And we see that when GLP-2 is administered during Golgi disruption by Berfeldin A, there's no effect on lymph flow, TG concentration, or TG to ApoB48. So this gives further support to this idea that GLP-2 is acting extracellularly, because blocking the intracellular pathway does not influence how GLP-2 can mobilize chylomicrons. So in summary, we see that both glucose and GLP-2 mobilize intestinal lipid stores in humans and animal models. And several lines of evidence suggest that glucose is mobilizing those intracellular lipid pools, while GLP-2 is acting on extracellular lipid pools. And we have a lot of questions coming from this work. So what we're trying to look at is, does glucose need to be metabolized to enhance chylomicron secretion? So our approach to this is to repeat those surgical model experiments, but this time using 2-deoxyglucose. So this is a form of glucose that gets absorbed into the enterocyte, but it will not get further metabolized. So we can see what the role of glucose metabolism is in enhancing chylomicron secretion. We also want to know, does endogenous GLP-2 enhance chylomicron secretion? So far we've just been looking at giving a high pharmacological dose, but we want to know what the actual physiological role of GLP-2 is in enhancing chylomicron secretion. So our approach here is to give intraduodenal lipid in the presence of a GLP-2 antagonist. So by antagonizing the receptor, what effect does that have on chylomicron secretion? And we think that GLP-2 is primarily acting on the lymphatic pumping, but to really know this, we want to see, does GLP-2 enhance lymphatic pumping by quantifying both the frequency of pumping and the amplitude or the strength of pumping in response to GLP-2. So with that, I'd like to thank Gary Lewis's lab, Chumting, Avital, Brenda, and Linda, and the Buman lab at Purdue University, who's doing the proteomic and transmissional electron microscopy work for us. And then our funding sources, CIHR, Diabetes Action Canada, and BBDC. So thank you, and I'll take any questions.
Thank you so much, Priska. So just a reminder, if you'd like to ask a question, what you can do is you can either raise your hand or you can use the chat room feature.
Do I stop sharing the screen now, or?
I think you can keep it on, that's fine, in case you need to refer to a particular slide. Yeah, so Dr. Sabowski has a question.
Okay.
Hi, very interesting presentation. I was intrigued in how glucose was able to mobilize lipid from enterocytes from the intracellular pool in enterocytes. Do you think that it would be possible that glucose would have similar effects on other cell types, like endothelial cell? Like, could it potentially affect LDL transcytosis of LDL through endothelial cells by modulating snares and thus trafficking of LDL through other cells?
I don't know too much about that, to be honest, but glucose does seem to be acting a similar way, like on snare proteins in the pancreas, for example. So it could be that this is sort of a broad effect throughout many tissues, that glucose affects snare protein assembly. But I can't say that I know for sure about LDL transcytosis.
Okay, is there another question from the audience?